So now I'm going to brief about the two main important binding sites of TSC2 that's going to interact with 1433, and that is S939 and T1462. And they confirm it by doing mutating both these sites, which we are calling the SATA mutant. So to perform this experiment, they actually reconstituted the wild type TSC2 cells and also these point mutants or the double mutant SATA into the TSC2 double mutant cells, and after that they performed immunoprecipitation to check the interaction between the proteins, that is TSC2 and 1433. As you can see, in SATA mutant there is no interaction between TSC2 and 1433. That confirms that these two binding sites, the arginine the serine and threonine S939 and T1462, are important for the interaction between 1433 and TSC2. You can also check it that either of the sites, when are mutated, you can see a little band, which does mean there is a little interaction, but when both of the sites are mutated, there isn't any visible band. So that does showcase the importance of the SATA mutant in this PI3K AKT dependent pathway also. This S1210 domain is actually a MK2 phosphorylated site, so it doesn't matter for the PI3K AKT dependent site, for the PI3K AKT dependent phosphorylation, because if this site is mutated, we can see there's interaction between TSC2 and 1433 when when TSC2 is regulated, when TSC2 is phosphorylated by PI3K AKT dependent pathway. They further also confirmed it with or without orthmenin. As you can see, orthmenin is an inhibitor of the PI3K dependent pathway. When there is the inhibitor, you cannot see any interaction because of no phosphorylation of TSC2. And you can see that when there is no orthmenin, you can see there is a PI3K dependent regulation which does phosphorylate the TSC2. That does mean TSC2 gonna inhibit TSC2 gonna bind with 1433. 1433 is gonna inhibit TSC2, and this TSC2 cannot inhibit the mTORC1 activity, and thereby you can see the downstream phosphorylation of S6 kinase here in hypoxic condition also. They further confirm it with normal and hypoxic condition. You can check that in wild type TSC2 reconstituted cells, there is no interaction between the 1433 and TSC2, and there will also no downstream regulation of S6 kinase because REDD1 is gonna inhibit 1433, so TSC2 is free, it can inhibit the mTORC1 activity. And in SATA mutant, you can see both in normal and hypoxic condition there is no interaction. That does mean this binding site is important. This binding site is important for the interaction between TSC2 and 1433. But you can also see some phosphorylation of S6 kinase. It is because that SATA mutant form have some reduced or altered activity in compared to the wild type TSC2 protein. So for SATA mutant to fully regulate the mTORC1 activity, and thereby the downstream signaling is compromised. Hence we can see a little phosphorylation of S6 kinase. But overall in SATA mutant, the binding of TSC2 and 1433 is not gonna happen. Now they showcase the importance of RPAA binding sites, the RPA binding sites of REDD1 that's gonna bind with 1433 and which is not gonna inhibit TSC2 to further downstream processes. So they confirmed it again with hypoxic condition and in normal condition with REDD1 and with REDD1 and without REDD1. You can see without REDD1 there is an interaction between 1433 and TSC2 both in normal and hypoxic condition, and there was subsequent phosphorylation of S6 kinase. That does showcase the importance of REDD1 in hypoxic condition. You can see in hypoxic condition when there is REDD1 there is no phosphorylation and there is no binding between, no interaction between TSC2 and 1433. For that the expression of REDD1 then further was induced with tetracycline. This tetracycline for 3 hours. This test was performed in osteosarcoma cells, and after that they performed Western blotting to check the interaction between the proteins, that is REDD1 and that is TSC2 and 1433. So you can check that in wild type REDD1 cells with presence of REDD1, S6 kinase is dephosphorylated, and when this RPA site is mutated, you can check that the that the presence of tetracycline doesn't matter because S6 kinase will be phosphorylated. That does mean the continuous activation of mTORC1 activity and subsequent cellular growth and proliferation. So this RPA site, which is a arginine and proline rich conserved domain of REDD1, that is actually interacts with the 1433. It's important in order to inhibit 1433. You can check the interaction between TSC2 and 1433 in wild type and RPA mutant. You can check that in RPA mutant there is interaction between TSC2 and 1433 with or without tetracycline. That does indicate that REDD1 cannot inhibit 1433. That's why 1433 is inhibiting TSC2 here. You can also they actually further confirm it with again the presence of insulin and orthophenine. You can check in wild type REDD1 when they put tetracycline, you can see no phosphorylation of S6 kinase. Although there is phosphorylation of AKT, but there is no phosphorylation of S6 kinase. That does indicate that there is no cellular growth or proliferation or absence of mTORC1 activity in presence of REDD1 in even they put growth factors like insulin in the culture. And also you can see that in RPA mutant, even though they put tetracycline, you can see there is a phosphorylation of S6 kinase and obviously there is a phosphorylation of AKT. That does indicate that mTORC1 activity is not inhibited by TSC2 because 1433 is gonna inhibit TSC2 here. They further confirmed the interaction, the co-amino precipitation by doing co-amino precipitation between 1433 and REDD1. Again, you can check in insulin and when there is insulin and tetracycline in REDD1 wild type cells, there is mild interaction between 1433 and TSC2. That does indicate that REDD1 is going to inhibit 1433. And in RPA mutant in insulin with tetracycline, you can check that the interaction between 1433 and TSC2 is much more. So that's why 1433 is going to inhibit TSC2, and that's why we can see the phosphorylation of S6 kinase here in REDD1 RPA mutant. They further confirmed it by doing experiments in normal and hypoxic condition. You can check that in hypoxic condition there is no binding. Even they put insulin, there is no binding, no interaction between TSC2 and 1433. And they further confirm it by the dephosphorylation of S6 kinase here. And in REDD1 wild type cells, they showcase the importance of these two sites of TSC2 that is already explained by my friends, that is serine 939 and threonine 1462. These two important sites are important for TSC2 to interact with 1433. So the conclusion, they told that hypoxia does the same as tetracycline induced wild type REDD1 does, because they actually inhibits the insulin effects on TSC2 and mTORC1 signaling. You can check that in REDD1 wild type cells with presence of insulin and tetracycline, the phosphorylation of TSC2 is always there. Means the TSC2 will be phosphorylated if there is any AKT signaling, because insulin induced AKT signaling. If there is any AKT signaling, the phosphorylation of these TSC2 sites will always be there, irrespective of the REDD1 interaction or not. I'm going to move to the next slides to brief about the binding more of this TSC2 and 1433 and why it is required even in hypoxic condition. So for that, again, the TSC2 double mutant mouse embryonic fibrous cells are actually stably reconstituted with wild type and this mutant SATA. This TSC2 actually co-transfected with REDD1 again, and to check the activity of S6 kinase. And after that, they performed the co-amino precipitation to check the binding and interaction between the protein. You can see. And again, I am going to tell you this del C is the deletion of conserved central domain of REDD1, which is also important for binding like RPA. As you can check here, when they put GST is a glutathione S transferase protein tag, which is actually fused with 1433 protein in order to check the activity. You can check that in wild type cells there is an interaction between REDD1 and 1433. But when there is a deletion of this conserved central domain or either this RPA domain which we just studied, you cannot see any interaction because these two sites are important for interacting with 1433. The further did a experiment with R18 decoy peptide, which is a competitor of TSC2 and REDD1 to bind with 1433. You can see that by increasing the concentration of this R18 GFP, you can check there is no interaction between 1433 and REDD1 because it is a competitor and it will compete with REDD1 and both TSC2. So you cannot see any binding between them. Again, when they remove that R18 decoy peptide and put normal GFP, you can see that the binding between REDD1 and 1433 is evident, and also the binding between TSC2 and 1433. Again, they confirmed it by putting by transfecting this flag 1433. Flag is the epitope that consists of 8 amino acids. It's fused with 1433. You can check when they put REDD1 expression. You can check the interaction between REDD1 and 1433. But when this is RPA mutated, you can see although there are more 1433, it cannot interact with REDD1 because the main binding site of REDD1 is mutated. Again, they performed it in wild type and SATA mutant of TSC2. You can check in SATA mutant when they put REDD1, there is phosphorylation of S6 kinase. In SATA mutant, you can check that when they put REDD1 in SATA mutant, there is a phosphorylation of S6 kinase, which means that this SATA mutant of TSC2 cannot actually fully inhibit the mTORC1 activity, and thereby you can see subsequent downstream regulation of S6 kinase. And in wild type cells also you can check the dephosphorylation of S6 kinase in presence of REDD1. This is important because they reconstituted the wild type TSC2 cells in TSC2 double mutant cells, and further they put REDD1 to check the interaction, and you can check there is phosphorylation of this S6 kinase, as there is a little amount of TSC2 is there, so we can check a hypophosphorylation of S6 kinase, and obviously with presence of this REDD1 is actually it's possible because you can check in SATA mutant or MUC mutant, the level of phosphorylation is high. Further, they performed experiments in they actually counts the viable cells in trypan blue exclusion assay, and they count the cell numbers and and formation of cell colonies under normal and hypoxic condition. And you can check by the graph that in normal condition, in wild type and SATA mutant, generally the formation of cells are similar in normal condition, but in TSC2 double mutant cells you can check that the cells are proliferating because mTORC1 activity is not inhibited, and subsequently there will be phosphorylation of S6 kinase and cellular growth and proliferation. But in hypoxic condition, this is important. You can check that in wild type cells the cells growth are in a controlled manner, but in SATA mutant you can check as 1433 cannot inhibit TSC2, and 1433 cannot interact with TSC2, and also that's why REDD1 cannot inhibit 1433, so that you can check that the cell growth or the cell number is higher in hypoxic condition after 4 days. And also you can see that it is more or less than the TSC2 double mutant cells. So they further checked it with colony number. So you can check in hypoxic condition the colonies are more formed in double TSC2 mutant and SATA mutant in comparison to wild type, because in wild type cells you can check both in normal and hypoxic condition that they nearly have a equal number of colonies forming under this normoxic and hypoxic conditions. And overall, if we talk about the percentage of cells that's gonna grow in SATA and MOC mutant is more high than in wild type. So now I'll ask my friend to brief about the REDD1 dependent hypoxic signaling. So now I'm going to ask my friend to brief about the REDD1 dependent hypoxic signaling that actually suppresses tumor genesis.